In our human bodies, there are a few ways that our immune system fights off invading pathogens. One of these ways is through adaptive immunity involving B cells and their interactions with other immune cells. Before we begin, know that every cell has unique protein markers called antigens on its surface. This includes our body cells as well as pathogenic cells. Our immune cells look for these antigens to determine if a cell is foreign or if the cell belongs in our body. We will be focusing on a specific type of immune cell, a lymphocyte called a B cell. B cells mature in the bone marrow and circulate in our bloodstream. These are the cells responsible for our ability to fight off an infection that we have had contact with and recovered from in the past. B cells function to make antibodies against specific antigens. Antibodies bind to an antigen at the surface of the invading cell, and in doing so they present the foreign cell to other immune cells to engulf or destroy them. We will discuss more about this later. So, B cells circulate in the blood and bind to the receptors on a foreign cell when they come in contact with the foreign cell. When this happens, the B cell is said to be sensitized. B cells have proteins on their cell membrane that are a part of a class of proteins called Class II Major Histocompatibility Complex Proteins, or MHC2 for short. These proteins allow for binding with helper T cells, specifically the T cell receptor proteins on the T cell membrane, which release proteins called cytokines after the T cell binds to a sensitized B cell. The pink dots represent cytokines and are involved in cell signaling, in this case activating the B cell. Once a B cell is activated, it either becomes a memory B cell or a plasma cell. Memory B cells are held in reserve for the next time the specific pathogen enters our system again. Plasma cells are the ones responsible for making and releasing antibodies that will bind to antigen sites of the pathogens. The shape of an antibody is important because we can see how they bind to antigens of foreign cells. Epitopes are regions on an antigen that bind with antibodies. A complete antigen refers to an antigen that has binding sites that allow both binding sites of antibodies to bind with one antigen at the same time. Antibodies play a vital role in helping other immune cells take care of invading cells. Antibodies inactivate antigens in a number of ways. In neutralization, the antibodies bind to areas on viruses and bacteria, so they will not be able to bind to other cells in our body, a process called adhesion. In precipitation, the antibodies bind to antigens, creating a soluble substance that is easier for phagocytic cells to digest. Antibodies also play a role in the complement system, where when the antibody binds to an antigen, it allows another protein from other immune cells to bind to the foreign cell and work to destroy it by ingesting the pathogen through phagocytosis. The small molecule hapten plays a role in the complement system as it can bind to complement proteins and also antibodies. Opsonization is the term for when a bunch of antibodies coat the surface of a pathogen in order to make phagocytosis easier for other immune cells.